Hello, everyone. Thank you for joining us for our webinar. Today, we are honored to have special guest host, Dr. Anna Belkina, the Assistant Professor, Department of Pathology for the Boston University School of Medicine. She's also the Associate Director of the Flow Cytometry Core Facility. In addition, Dr. Belkina is an ISAC SRL Emerging Leader for 2015 through 2019. I'd like to introduce our Senior Application Scientist, Chenjun Zhang. She'll be hosting the webinar today. Take it away, Chenjun.
Hi, thank you, Liz. Hi, everyone. Thanks for joining the webinar today. This is Chenjun Zhang. We are honored to have Dr. Anna Belkina with us. I'd like to say that Anna and I both share deep roots within ISAC. As Liz just said, she's very active within the society and she's a SR emerging leader there. Her last webinar with Cytobank featured Visni was very popular. We had record high attendees a few years ago. You may go to Cytobank YouTube channel to watch the recording there if you haven't already. For today, I will give an overview of Cytobank, introducing some of the tools available to help visualize and analyze high-dimensional cytometry data with a focus on citrus. Dr. Belkina will then share her research on how she used citrus to identify stratifying features that differentiate HIV positive and Asian cohort versus uninfected controls. It will be followed by live Q&A. You may submit your questions anytime during the talk in the Q&A window. For those who are not familiar with Cytobank, it's a cloud-based single-cell analysis platform for high-dimensional data. As nowadays, with the advancement of technology, we are able to measure many parameters simultaneously on a single cell level. You can do 10, 20, 50, or even more with some of the genomic techniques like CytSeq or RNA-seq. So also the number of samples increased dramatically, especially in the context of clinical trial, where different patient cohort, different treatment and time points are measured. So the whole field is going toward high dimensional and high throughput. This imposes huge challenge on data management, data analysis, and visualization. Also, the data reproducibility with cytometry data is also a concern. We all know there's quite a bit of expert variability associated with manual gating. So people seek help from computational algorithms, which are very compute intensive and require scalable computing power. Also with big data, transfer and share the data among collaborators globally becomes a challenge too. So Cytobank comes in with the cloud-based solution to specialize in high-dimensional data so that we can leverage cloud computing power for many of the algorithms we have in Cytobank. We also offer structured content management where you can securely save and archive your analysis. You can collaborate and share easily with colleagues too, just by a share button. So the platform is designed for biologists. No coding is required to run any of those algorithms. You can also make Cytobank's professional bioinformatics consulting team as part of your team for your research project. And we welcome any potential collaborations in that regard. Here's a list of the machine learning algorithm available in Cytobank. We have clustering tools like SPADE and FlowSum. FlowSum is one of our new release that enables you to cluster more events in shorter time. Visni, you may all familiar with, it's the dimension reduction tool allows you to map n-dimensional events onto 2D plot and still preserving all the information of all events. Citrus is our focus today. It's a powerful statistical tool to differentiate the differences between your sample groups. It's probably one of our most complicated tools. We'll be back to that in more details in later slides. Before we jump to Citrus, I'd like to introduce some of the key functionalities in Cytobank. We have a whole suite of data analysis tool We see data analysis as a pipeline workflow. The first step will be data pre-processing, which includes compensation, scale transformation, and pre-gating to get a clean quality data for downstream analysis. After that, you can either use clustering tools or dimension reduction tools like Visney, Frosum, Spade, and Citrus. As we said earlier, you can leverage the cloud computing power to run multiple analyses simultaneously and free up your local computer that way. There are a few other features in Cytobank that allows you to extend the functionality further. Job is the feature allows you to convert non-cytometry data to FCS format so that you can analyze them with all the tools available here. Or you can run command line with Cytobank API, the application programming interface, to interact with the data stored in Cytobank. So normally, user access the data use web browser for Cytobank. You can gate your samples. You can run Visney run requests to the cloud within your browser. But the API really extends the functionality where you can send, batched instruction to batch upload, download data to Cytobank. You can also use this interface to communicate with the electronic lab notebook or LIMS system. So the communication, transfer, and data information between those systems become an automated process. Or you can even use customized code to build a pipeline to run algorithm that's otherwise not available within Cytobank. Then you can upload the results or the data back to Cytobank. So the API allows easy collaboration between bioinformaticians, statisticians, and biologists. Cytobank can serve as the central platform connecting all those workflows. Here, as we said earlier, job, you can convert any tabular data into FCS format. It can be a CSV file or text file. You can upload that file normally into the upload data interface within Cytobank. You may choose the column and rows to be included in the data. Then the job will convert that file into a FCS format. So once it is converted to FCS, you can use all those tools that's available, for your CytSeq data or your imaging data and some of the other single-cell genomic data. Here's an example showing what the CytSeq data looks like in Cytobank. With some filtering and normalization, the events looks quite similar to conventional cytometry data in this 2D plot. You can also visualize the data with the Visney algorithms as well. We just had a webinar last month with Dr. Deep Roman. He featured his CytSeq analysis workflow within Cytobank. You may check it out with our YouTube channel. So finally, back to Citrus. So what is Citrus? Citrus stands for cluster identification, characterization, and regression as a names. It has several components associated with it. First step will be unsupervised hierarchical clustering to identify the phenotypically similar cells in all samples. Then it will go to characterization of those clusters by either abundance of subsets or median expression markers like activation marker or phosphoprotein markers. And then statistical models are used to identify the stratifying biomarker that can be used to tell the differences between groups. Citrus is a very automated process where all the process here is within the black box. You start with your scientific question and end with stratifying biomarkers. So put it in a simple way, basically it is used to identify significant differences that can differentiate groups. Citrus can help you to perform unbiased and potentially more thorough data mining and inspection of cell subsets within your data. The statistical models with Citrus can be correlative or predictive. Correlative model can be used to identify all the significant differences in cell populations between sample group. And there are two predictive models, PAM or LASSO. We'll talk about those models next. Here's another slide showing, which model should you choose once you're running Citrus. It depends on, really, as we said, depend on your questions. If you're trying to identify all the significant differences between your groups, we may run SAM, the correlative Citrus. Or if the question is to identify the minimum number of features that are necessary to predict the endpoints, like which sample belongs to which group, you may run the predictive model. An example of that will be, cancer patient treated with immunotherapy, where if we are trying to identify all the differences between the responders and non-responders, you may run SAM. Or if you're trying to identify, what is the minimum number of biomarker or cell subsets that can tell the difference, which sample belong to the responder group or non-responder group, we can use the predictive model. And there are two models associated with the predictive stages. One is a PAM and one is LASSO. There are a few differences between them. LASSO can only run with two groups, and there's no false discovery rate associated with this model. If you're running with more than two groups and false discovery rate is designed, then you should run PAM. So in Anna's talk later, she used both models. Predictive model was used to identify the minimum number of features necessary to predict those HIV positive and non-infected controls. Correlative model was used to identify, all the clusters that are significantly different between those two groups regard to differential inhibitory and receptor expression. You'll hear more from her in a moment. So we mentioned earlier that is a cluster characterization process involved. After you decide on which model to run with Citrus, you now can choose, which feature you would like to run. The features include either median expression of markers in your panel or abundance of certain subsets. You can choose one or another with one Citrus run, but you can set up two Citrus runs to check on both. Again, when to use what is also depend on your study. If you are hypothesizing that there are biomarker expression level change with your patients receiving cancer therapy, such as immune checkpoints, PD-1 expression level within your responder and non-responder, you may choose the median. Or if you are looking at change of frequency of certain cell type, like Tregs with different disease status, some patients are recovering after the treatment, some patients are still progressing with the disease. So you may use abundance for identify the frequency of subsets or clusters. Here's an example showing, the setup of Citrus. First of all, you need to have at least two groups to start with. Assign your samples to different groups based on different outcome or different endpoints, like responders or non-responders. Then you run Citrus to identify clusters of phenotypic similar cells in all samples. And once the clusters identified, we'll start to characterize the biological features of those clusters on a per sample base. Citrus then use either correlative or predictive model to identify the subset feature that best predict the endpoints, or use correlative model to identify all the differences between two groups. So here is a cluster, this particular cluster discovered with differential phospho-S6 signal in this particular cluster, and between those group A, responder group, and the non-responder group, we can then use to identify this cluster further after run Citrus. Here's a Citrus tree looks like. This Citrus tree, starts with a central cluster that contained all events from all samples. Then it branched out with child and grandchild clusters, and each parent cluster contained the events of its children. And there, you probably couldn't see, but there are arrows between the cluster pointing from parents to child. If the connected clusters are identified as significant, as highlighted here, you probably can just pick the parent cluster as it cleans all the child cluster here, unless you want to identify the subsets in more details. So in this case, a cluster was identified as differences in the abundance with those two groups. And then you can check on the marker expression on this particular cluster to identify the phenotype. So other than, you can run Citrus by itself. You can also incorporate Citrus into a data analysis pipeline with other algorithm like Visney. So in this workflow, you can run Visney first, and then you run Citrus on your Visney data, so that your Citrus results will have this T1 and T2 channel, where you can now overlay your Citrus cluster back to Visney to identify what's the phenotype and what they look like on the Visney map. I'll show you an example here where Citrus was run to identify the median expression of this phospho-S6 expression in the control and the BCR stimulated samples. Then you will see, once this cluster is identified, you're able to, this is a Visney map of the same data. You can able to overlay that cluster back to your Visney map. And once it's on the Visney map, you're able to, highlight with the third channel to check on the expression marker of, some of the lineage marker like CD20. You'll see they are mostly significant cluster is belong to B cells. And also you may also turn on the phospho-S6, which is not showing here, to see the expression level of that particular cluster. So here I just showed you options where you can, how you can use Citrus and identify, and some of the data visualization and interpretation tools within Cytobank. If you have any question, I'll be happy to take it during the Q&A session. But without overdue, I would like to turn the screen for Dr. Belkina's talk. You will see, how Citrus was applied to her study and making unexpected discovery in the HIV and aging cohort. Anna, it's all yours.
Thank you. Thank you everybody for joining. Thanks Chanjung for your introduction and overview. So I'll just jump into the story, just not to lose time. So our team has broad interest in how the functional activity of immune cell populations is altered in chronic diseases. And specifically, we're interested in cells that bridge innate and adaptive immunity, which include natural killer cells, natural killer T cells, another new T cell population called gamma delta T cell. So when the immune response begins, the microbe or tumor antigens are infected or taken up by the cells in the microenvironment, such as an immune cell, innate immune cell depicted there. It can be a dendritic cell or macrophage. So the cell will then respond to the new antigen or infection by changing the MHC expression, and that will activate NK cells. Also by secreting some inflammatory cytokines, which would activate NK cells as well as NKT and gamma delta T cells. So the cells then secrete more cytokines, and either inflammatory or immunosuppressive, and they can also become cytotoxic. So the thinking is that the effector functions of the cells then dictate the nature of the memory T and B responses, and the cells are likely very important in the chronic disease, but overall they're understudied in comparison with traditional T cells and B cells. And conversely, the function of these cells is prone to the phenomenon of exhaustion. So what is immune exhaustion? Exhaustion occurs when specific antigens, such as from a pathogen or tumor, and or non-specific inflammatory factors are sustained in a host well past the acute phase of the response. So exhausted cells are believed to progressively lose of ex vivo functional capacity over time, and also upregulate so-called inhibitory receptors. And this process has gained a lot of interest in recent years, and I'm sure you're all familiar with that because due to groundbreaking clinical trial results using therapeutics that block the inhibitory receptor signaling, such as PD-1, PD-L1 axis, for example. So here's an example from a melanoma study where a patient was on anti-PD-1 clinical trial, and it shows complete eradication of the tumor 90 days after the initiation of the treatment. So this result suggests the reversibility of the exhaustion state, and this reversibility has been shown in animal models as well. And one view of how immune exhaustion progresses is that an individual cell will upregulate more and more inhibitor receptors over time, and this will be linked to the loss of effector function capabilities. However, evidence from our group and others indicate that this process likely occurs in a complex branched manner, with factors in the microenvironment driving distinct immune cell subsets into a variety of inhibited states. So we hypothesize that the states may be reflected through the individual inhibitor receptor signatures on immune cells. And we predict that inhibitor receptor signatures can provide new insight into how checkpoint inhibition progresses and how it can be broken into stages, what certain functions are lost, and how also would help us to determine at what point we cannot rescue a cell from immune exhaustion because it's no longer reversible. So back in 2017, we designed a 16-color flow cytometry panel that would allow to identify multiple lineages of immune cells and characterize their inhibitor receptor signatures all in one panel. So this panel was specifically optimized for our 16-color FACS-ARIA sorter to allow simultaneous sorting of multiple populations of interest. So we worked out several combinations of inhibitor receptors that would be relevant to the disease of interest, and all these panel variations would share the same panel backbone that allows to distinguish the major immune subsets. We applied this panel to a good half dozen studies by now, I think. And today we will be talking about our work with the immune exhaustion signatures in our HIV and aging cohort. So the question that we asked in this study was, is immune exhaustion of particular immune subsets linked to HIV and or age-induced so-called inflammaging? So to answer this question, we teamed up with Dr. Nina Lin, an assistant professor of medicine in our school, who has been working with this cohort for many years. So just to set up a backdrop for the story, people living with HIV have seen great improvements in both the length and quality of their lives in the last several decades, or at least last decade, since antiretroviral therapy now works very well at lowering and keeping viral loads below the limit of detection. However, even without detectable viral loads, HIV is still significantly detrimental to one's health, and such individuals are at increased risk for what is called serious non-AIDS events, or SNAs, which include cardiovascular disease, neurological problems, diabetes, cancer, osteoporosis, liver cirrhosis, frailty, pneumonia, and some others. So interestingly, these diseases are typically associated with aging as well. So there are reports of higher rates of these comorbidities in HIV-positive subjects compared with the general population at the same age. And there are some reports that these events occur in younger ages in HIV-positive people compared to uninfected controls. So this is the concept of inflammaging, that the very low levels of the virus that remain are causing inflammation that is somehow aging the person to develop this age-associated illnesses. And there's a debate in the literature as to whether HIV-positive persons are aging earlier or differently. And a key point to keep in mind is that it's not clear if these conditions in HIV patients manifest due to similar processes as what occur in normal aging, or via completely distinct mechanisms. What is well established in the field is that the occurrence of these non-AIDS events is strongly associated with plasma markers of general inflammation, including CRP, IL-6, soluble TNF receptor 1 and 2, soluble CD14, as well as some coagulation markers like D-dimer and fibrinogen. There was a study that followed HIV-positive patients over time, and it found that higher levels of these biomarkers before the occurrence of a serious non-AIDS events are present. And also the same plasma markers track with disease onset in non-HIV aging as well. So our questions were, first of all, is the inhibitor receptor expression of a particular immune subsets different with aging, HIV infection, or both? Does HIV infection and older age impact our immune measurements in an additive or a synergistic manner? Or do people with HIV simply immune age earlier than the general population? And finally, does expression of inhibitor receptors, or they called also immune checkpoints, always track with the loss of functional activity of immune cells? That's the definition of immune exhaustion. So the cohort that we use, called the HIV and aging cohort, was established by Dr. Nina Lin. And that cohort includes the 4 groups comprised of healthy young, healthy older, HIV younger, and HIV older subjects. We have over 20 subjects per group for most of our analysis. And the subjects were matched for sex and ethnicity. And also it's important to mention that the HIV negative subjects in the cohort have similar lifestyle and socioeconomic exposures as the infected subjects. So all the HIV positive subjects in the study have sustained viral load for a minimum of 1 year. And we collected PBMC and plasma for these patients and non-infected subjects. And we stained them with inhibitory receptor signature panel, collected fluorescence cytometry data. Also, we sorted multiple cell types from this study. And we had several biologically plausible hypotheses to test. And we wanted to use some unbiased analysis to go through those hypotheses and actually mine for new hypotheses. And here we go to citrus. So we fed all our flow cytometry data to citrus, and Chunjung has already walked you through the general outline, so I'll just focus on the specifics of the study. So you know what citrus is, that algorithm designed for a full automated discovery of statistically significant stratifying biological signatures in single cell data sets. So in our case, flow cytometry data. So we would associate the phenotype with some endpoint. In our case, this would be the subject group. And we carried all our citrus analysis in Cytobank. So assigned our subjects to groups per their HIV status. We also ran some 4-way comparisons. And also we tested if we can use age alone for subject classification. And we identified lineage parameters for clustering that are like a logical conclusion out of the panel design that we implied. So citrus is based on hierarchical clustering, as you've heard, would randomly select an equal number of cells from each sample, combine them, and cluster all at once. So what's interesting here is that all clusters in this tree would be considered for analysis, not just the peripheral smallest clusters, right? And that way, citrus would avoid the problem of calculating how many populations exist in this database. And that's kind of one of the main problems with most clustering algorithms. So the tree that you see here has a lot of redundancy because the same cell is included in multiple parent-child clusters. And you can expect that clusters that contain same cells will correlate with the same endpoint. But citrus algorithm modeling actually corrects for correlating features, and that is not a problem to calculate significance of your results. So we use modeling approaches that are built into citrus pipeline and to find statistically different signatures in our cohorts. And you know that citrus can assess the proportion of sample cells in each cluster and also the median level of each functional marker in a cluster of cells. So there can be several scenarios here. Scenario A would be that the cluster size based on lineage marker is significantly different between the groups. So for example, group A has higher percentage of CD4 cells than group B. The other scenario would be that non-lineage markers are differentially expressed between the groups. So for example, group A has more PD1 on the CD4 cells than group B. So we considered both scenarios in our analysis. And before I show you our results, I'd like to discuss our workflow. So we take our raw flow cytometry data, fluorescence data, and we apply compensation either on acquisition or using a post-analysis compensation platform. Mostly we use compensation on acquisition because we also sort the cells. So we do some quality assessment using both some automated tools and expert guided analysis. And then we usually transfer at this point data transferred to Cytobank. So that line indicates Cytobank position. And then we verify the correct data transformation, which is what you find under the scales setting in Cytobank. So we usually use arcsign transformation for all our fluorescence cytometry data. And usually the automated parameter selection works pretty well for us, but sometimes 1 or 2 channels they need adjustment. So it's really important for pretty much any platform that you use in Cytobank that you verify your scaling. And with flow cytometry data, sometimes scaling can be quite different between channels, so it needs to be controlled for. So then we identify subsets for citrus analysis. So in the default way, we would just use all our live lymphocyte cells for citrus. So you know that minimum cluster size in citrus is defined by user, And setting this parameter to small number will allow smaller, more rare cell subsets to cluster cleanly. But that will also reduce statistical power to detect associations. So if you know that your data set contains both large but roughly uniform populations and small populations, it may be tricky to get one run of citrus to compare all subsets. So if your minimum cluster size is large, you risk missing smaller populations. And if it is small, you risk having not enough statistical power. And in that case, also citrus will attempt to break your large populations into smaller but biologically meaningless clusters. So in our case, we had actually to subsample the smallest populations, which are iNKT cells, and they're under 0.1%. So we subsetted them out and ran them separately to reach reach sufficient granularity in our analysis. So that approach would introduce some bias to the study, and that's unavoidable, unfortunately. But we cannot cover everything with one parameter selection, right? So, and as always with any modeling, we try to verify our citrus results with alternative approaches. So we don't take it from citrus straight into submit to nature button, right? So citrus offers several modeling approaches, and they were outlined by Chin-Jung, and this slide is more like a placeholder for those who would be watching this later for studying. And I also highly recommend a webinar by Cytobank senior application scientist Geoff Cracker on YouTube channel by Cytobank. So that one covers the modeling in very much details. So in our hands, we found the PAM prediction analysis for microarray modeling the most helpful. And also these results agree best with other statistical analysis that we perform outside of the citrus pipeline. There's another predictive model, Lasso, and in our hands, we see that it has a tendency of selecting the smallest clusters as predictors. And since hierarchical clustering in citrus sometimes is a little bit sloppy, the results often look like we're pulling answers from overclustering or low specificity clusters. So we found the Lasso method less useful in this regard. And then also there is SAM, so a significance analysis of microarrays. It's a correlative model. It's also has its own niche. We find that it often seems to be a little bit overexcited with our data. So even if we set the most stringent cutoff for significance, and we would often use this modeling as a first pass for analysis, and also for finding possible batch effects and other artifacts in our data. So let me illustrate this. So this is some negative results that you usually don't see in publications. So from a different study. So we compared cohorts that were pretty much identical. And you can see that none of these PAM models do any better job than a random 50-50 guess. So this red line is hanging around 50%. We compared abundances, we compared inhibitory receptor median expression, we tried normalizing the scales or not normalizing the scales, and the result is pretty much the same. And that's expected. However, when we look at SAM results, SAM actually found several clusters to be significantly different in these two cohorts. And one of them, the CD8 one here, was just a pure artifact of overclustering. And another one, the CD3 cluster here, was not statistically different when this population was identified by other methods. But the third one identified a population that almost fell under the minimum cluster size limit, and that's why it was not picked up by PAM. So when we lowered the limit enough to get that population in, there was so much overclustering that PAM didn't find it significant and interesting enough. So we took this population and actually followed it up with other methods to check if there's a real biological phenomenon behind this result. So now going back to our HIV study, with real results, we clustered our data with citrus and assessed median expression of all inhibitory receptors. So in this batch of runs, we identified all major subsets of immune cells that we could see with this panel. So we can see CD8s and K cells, CD4 cells, and I don't think I mapped them here, but also Tregs. The exception was iNKT cells. As I said, they're distinct but very rare. So citrus couldn't cluster them cleanly. And with PAM modeling, the minimal set of features that distinguished our subsets was that the TIGIT expression solely on gamma delta T cells stratified the HIV positive subjects from non-infected individuals. This result was very robust. We reproduced it multiple times. And so just if you're interested in how we set up our runs here, we tried normalizing the scales and not normalizing the scales, and we didn't see much difference in this specific data set between these two set of settings. Now, here is more details about this. This is TIGIT expression in each gamma delta T cell cluster identified by citrus. It's consistently higher in the HIV positive individuals compared with controls. There's also some interesting heterogeneity in the gamma delta population that when we check out other surface markers expressed on this cell, such as CD8, CD16, CD56, CD127. And this is when you may want to subsample just gamma delta T cells from your data set into a separate citrus run to be able to find smaller but biologically meaningful subsets that would be too small for the cutoff of your larger run. Because as you can imagine, gamma delta T cells in blood would be probably less than 5%. So if you're subsetting them further, it's really hard to get clustering done cleanly with the universal cutoff. And when we look for FDR constrained models, so this is not a minimal model, not a minimal set of settings, but still significant set of settings. We find that two other inhibitor receptors, CD160 and TIM3, also differentiate our groups of subjects, but also only from the clusters containing gamma delta T cells. And it is important to note that inhibitor receptors are expressed across all subsets of immune cells in this data set. Finding the difference just in gamma delta T cells, whose role in HIV is totally unclear, and not in CD4s or CD8s, was very surprising for us, okay? And I mean, of course, we expected that we'll see the CD4-CD8 ratio shift in HIV individuals, which is a hallmark of HIV infection. And that was indeed reflected in our citrus results. So when we did abundance run, we found that we have change in CD4 and CD8 percentages. And just an anecdote, you can see that I have the SAM and the PAM models here. And you can see that SAM actually finds more clusters to be significant. And that goes back to my discussion about why we prefer PAM for more stringent results. I don't know if I can say it that way, but I would say SAM is definitely putting in things that we not necessarily follow up as significant by other methods. So next, we wanted to determine if this higher inhibitor receptor expression meant that the gamma delta T cells were actually functionally impaired. Were they exhausted or not? And we sorted all the gamma delta T cells from the blood samples, added them to small culture wells, and then after 20 hours collected the supernatant and measured 33 different cytokines in the soups. And interestingly, we again were very surprised by our gamma delta T cells. So on the x-axis here is the percentage of TIGIT positive cells from this subject sample. And on the y-axis is the average amount of different cytokines secreted per cell in the culture. And for the uninfected controls in the top line in black, it looked like we would expect. So the more TIGIT positive cells in the culture, the less cytokines they make. So the exact opposite was found in HIV positive subject, with significantly more cytokine produced with more TIGIT positive cells in culture. And the cytokines included soluble CD137, granzyme A, granzyme B, MIP1 beta, MIP3 alpha. We also saw some strong trends for perforin, TNF alpha, and interferon gamma. So TIGIT here is not an exhaustion marker of gamma delta T cells in the context of ART-suppressed HIV infection. So their functionality is clearly different in HIV positive and HIV negative subjects. So we can't call these cells exhausted anymore just based on the fact that they express an exhaustion marker. And at this point, we went back from our in vitro system to the patient data, put together our inhibitory receptor signature data with measurement of 16 markers of inflammation and cardiovascular disease in our subject plasma. Because remember, I said that there were biomarkers of inflammation in the plasma that track with the cardiac events and other comorbidities of age in HIV positive patients. So we have collaboration with Laufenberger's lab in MIT. So two postdocs from that lab, Olena Starchenko and Elizabeth Proctor, ran PLSDA, partially squared discriminant analysis on our data set. So with PLSDA, just like with citrus, you tell the algorithm who is in which subject group. So in our case, we had 4 groups: healthy younger, healthy older, HIV younger and HIV older groups. And the goal of this method is to determine if the groups are actually different, and if so, to determine which of the parameters you fed into the model are actually defining the differences in the groups. And here's the result. So PLSDA projects the multidimensional data into latent variable space, and each subject is color-coded according to their group. So you see healthy younger ones are green, healthy older are blue, HIV younger red, and HIV older yellow. We can see that HIV status tends to separate groups across the latent variable 1 axis, the horizontal axis, and the age translates into latent variable 2 axis separation. And all 4 groups separated pretty well, as you can see, and also as you can quantify, they're significantly separated. And a reasonable question that we asked ourselves before the reviewers would ask it, right, was what if the plasma data alone would do this separation? So we did the same modeling, but replaced the gamma delta T cells, T cell signature with CD8 T cell inhibitor receptor signature. So CD8 cells also express all 4 inhibitor receptors. And they were not different by citrus. So when we did this replacement, the model failed to separate the groups. So we were pretty confident to say that gamma delta T cells are integral to this 4-group different inflammatory states, and that HIV younger subjects are clearly separated from healthy older individuals, meaning that HIV infection is not simply aging your immune system. So when we have aging and HIV infection collide, that creates a completely new inflammatory network and process. Gamma delta T cells are likely a component of this divergent inflammatory process, aging without and with HIV. So the bottom line is HIV does not simply induce early aging. So the take-home messages from this study is that we use citrus to analyze inhibitor receptor signatures on different subsets, and we revealed that TIGIT on gamma delta T cells exclusively defines aviremic HIV positive subjects from controls, therefore may be an effective biomarker or therapeutic target. TIGIT on gamma delta T cells is associated with HIV, with activation of the cells in HIV positive subjects and not with exhaustion. So you can't use inhibitor receptor expression as a universal correlate of immune exhaustion. And finally, with our modeling, we revealed divergent aging process with and without HIV, and likely gamma delta T cells are a component in this inflammation process. And with that, I would like to thank my collaborators, Dr. Jennifer Snyder-Caponi, here with whom we run this project, Nina Lin, our clinical collaborator, our MIT collaborators, and of course our Cytobank collaborators, so Kat Draker and Chris Cicolella, who are now who left Cytobank recently, and then also Jeff Cracker, who is still with Cytobank. They all helped us a lot with forming and completing this project, and we're looking forward for further collaboration. And of course, I always thank Isaac for the support of my research. Thank you.
Thank you, Anna, for the great talk. Let me transfer back to my screen now. So we're going to start with the QA session, but just to summarize that, if you are new to Cytobank, please sign up for our 30-day trial on the Premium. And you can also take advantage of our training and consulting services. We are happy to provide any consultation on your project. We also have great educational resource. As Anna mentioned earlier, we had a Citrus YouTube video that shows, explain all the models in more detail on our YouTube video. And also, we just recently released a Flowsum blog post by our scientific team. You're welcome to check them out. We have very strong support and help portal. We can provide live troubleshooting on your experiment within your account. So yeah, just get in touch and let us know if you have any questions. Now we're going to start with our QA now. I see the first question coming. It says, so in order to overlap citrus cluster on vicinity analysis, how is the workflow? First vicinity and then run citrus? Yeah, this seems to be for Cytobank. I will be happy to take that question. So yeah, there's a few extra steps if you are trying to overlay your citrus, identify the citrus cluster on your vicinity plot, first you will need to run Visney first on your citrus data, on the data you're trying to run, finding the different biomarker. So run Visney first, then you can run citrus on the Visney channel you use for the clustering. And then as a normal flow, once the clusters are identified, there's a few extra steps. You need, concatenate the files and then upload them back to the original file. So yeah, if you need to know if you have any question during your actual data analysis, you can write into our support at cytobank.org. I'll be happy to provide step-by-step instruction that way. I hope that answers your question, and let's see if there's any more question coming. Okay, actually, Anna, I'm going to ask a question on behalf of a lot of users here. I know a lot of people are interested in, as you have already published the paper, if the data associated with the publication is available to the public access, and if so, where can they access the data if they want to try out citrus on themselves?
Sure. So I have this data are all loaded in Cytobank. I am happy to share the experiments with you if you just look me up and send me an email and tell me what your handle is in Cytobank. I'm happy to share the experiment that contains our data.
Oh, that would be great. Thank you. Yeah, so for Cytobank, we do have the option to make the experiment public if you wish so. So we also have some public data that you can test on citrus on. So just sign up with our premium. You will be able to access the citrus dataset, demo dataset we have now, and you can try out all the functionality yourself. Let's see, I do have another question here from the attendee says, how to decide the number of clusters when running citrus? Is there any approach that could be indicative, or do we need to test different values and check which one shows better results?
Sure. So you do not directly tell citrus to say the number of clusters. It's not same as, for example, for spade, where actually you give it a number. So in citrus, instead, you set up the percentage of population to be clustered into the smallest cluster. So your smallest cluster will have at least that percentage of your total population. And that number should come from some educated guess. So if you know the heterogeneity of your population and you have some general understanding of what it would be, you can certainly use that as an initial number. You can also test different values and see the results that you're getting. In our case, for example, we have a big population of CD4 and CD8 cells, but we really do not expect to see a lot of subsetting of those populations with the markers that we have in the panel. So we tend to overcluster them in the runs where we have we're aiming for the smaller subsets. So this is often the question that you need to answer in different ways to assess comparisons in different populations.
Yeah, exactly. Thank you for that, to answer that question. So actually in Cytobank, when you set up citrus, you're able to identify the smallest cluster you're going to define. So you could set up a few, then you can run them simultaneously and check on the results that way too. So I do have another question for Dr. Belkina here. So he asked that in your workflow after compensation and before getting into the steps using Cytobank, you have the quality assessment step. What are some of the QAs that you do? Do you have a checklist of these?
So some of those steps are basically outlined with all our procedures with general generation of flow cytometry data, and they include the quality assessment of the instrument. So we usually review that all before we do any analysis of the data. We also look for batch artifacts using some analysis templates that we mostly do in Flojo. There are also several scripts that would automatically scan for irregularities in your cytometry data. So we use several R packages for that, and also some Flojo plugins. So there's so the scripts, some of the scripts would be Flow Clean. There's also Flow AI. So we usually run a batch of them and use some kind of a compendium of those to make decisions whether some of the samples should be excluded or some artifacts should be cut out of the recording. I mean, the samples are often pretty large, so we have hundreds of thousands of events, and we can afford excluding some if there's some irregularity with the recording. So that's just part of the normal QC procedure. We also sometimes, the artifacts become more clear when you actually do algorithmic analysis. So if you have some batch effect, you will see, for example, that some of your samples tend to cluster into specific parts of the tree in your citrus run. So that would clearly indicate that you have batch-to-batch variability.
Great. We do have another question for Cytobank. He said, I'm a new grad student and the only person in the lab doing high-dimensional flow. Does Cytobank have education promotion for individual students? Yeah, thank you for asking that. As I said earlier, we offer a lot of education resources. We have the Cytobank YouTube channel with all the past webinars, also basic training videos. If you are interested in get a personal touch, just write in to us, and we will be happy to set up a demo for you. That way to get you started. But otherwise, we have great online resource and education with our support articles. Also, a great way to get started. Let's see, here's another question. It says, what was the phenotype of the gamma delta T cells between the HIV and normal patients, young and old? That's for you, Dr. Belkina.
So yeah, I think I answered that privately. So we're actually investigating this right now. We're working on a new study where we have a larger panel that characterizes gamma delta T cells in more detail. So I can say that these phenotypes are very diverse. So we see not a single population of gamma delta T cells, but at least 5 or 6 different subsets. So that we couldn't really characterize in detail enough in this study. So we endeavored another study that we're working on right now.
Got it. Yeah, so another question that I would like to ask is that, once you have the citrus, I think one of the slides you show, both SAM and PAM, and definitely SAM found a lot more significant clusters as it is intended to do because it's finding all the significant clusters. So I want to see, have you follow up on the significant difference that was identified SAM, but not the PAM? Did you do any manual verification or any other models to identify, if they are, significant or?
Yes, and also there's another question that asked like how we detect artifact clusters that kind of relates to the same thing, right? So yeah, we never trust one method, and it's not that we discriminate citrus for some reason. Like we never trust one method when we do any data mining, right? So citrus uses hierarchical clustering, which sometimes has its own drawbacks in terms of the cleanness of the clustering. So we would always do some alternative tool, and we use FlowSum a lot, for example, and we used to use Spade more before, and then we basically want to verify that the same population can be either gated manually or clustered by other methods, and we can actually find the cells to be sure that we're actually seeing something real. So that would be one thing. And then we would investigate if the cells cluster with multiple algorithms. We would actually investigate what the phenotype is. And most likely it would be that the SAM picked it up because of the threshold being slightly different, but actually if there is a biologically plausible explanation for the difference, then most likely it will be picked up by other methods as well. if it's more like artificial thing that we only find in citrus, we would consider this basically our over mining, and we would not take this into account in the future runs. So we never just rely on one method.
Got it. I do have another, probably that will be our last question due to the time. So I have a question asking, is there a minimum number of events per sample that a citrus analysis can be performed? Example, minimum 100 events per sample in a group of 20 individuals. Anna, do you have any insight on that, or otherwise I can take that one?
Well, I'm not exactly sure how it's implemented in Cytobank. So I know that technically you can go, I think, very low. Practically, you come into the question, if your cluster has only like 5 cells, is it something meaningful that you're looking at, or completely not? I think the cutoff is 50 cells per cluster in the implementation of citrus that I used last time. And that's kind of what we tend up when we looked at the iNKT cells. That's probably the cutoff that we set, because those cells are very rare.
Yeah, so actually that's exactly with citrus run. For the Cytobank setup, you can go very low. You can put in the equal sampling. We can go very low. However, as Anna said, it need to be biological, making biological sense. If you only input 100 events per sample, then it probably doesn't. The significant finding probably doesn't make any biological sense. So it really depends on your experiment setup. If you are using other genomic data other than cytometry data, probably you could go very low to run the citrus. So yeah, we can discuss that more. If you are have further question regard to this, just write to us to support@cytobank.org. We'll be happy to take that discussion from there. But otherwise, well, thank you so much. Thanks everyone for attending the webinar today. It was a great one, and hopefully you learned a lot about citrus, or maybe you have more questions about citrus. So welcome to send any question to us or to Dr. Belkina. I think she has her email address in the slide over there. So Dr. Belkina, thank you so much for your time and effort to put in such a awesome presentation. So thank you.
Thank you all, and we will be sharing my slide deck. So guys, if you're interested to review it again, it will be posted by Cytobank shortly.
Yes, we will also send the video recording to your sign up email. So look out for that too. All right, see you next time, everyone. Bye-bye.
Thanks, everyone.